Hello, I am going to present our work, Classification of 3D Interpolated EEG Signals using Hybrid Recurrent 3D CNNs. Electroencephalograms, also known as EEGs, are the recordings of the impulses emitted by the brain. The different hemispheres of the brain interact with each other in specific patterns to perform certain tasks. The readings of the EEG signals from the various parts of the head can be recorded through the skull for a specified time frame and extracted to give valuable information. These readings are collected with the help of an EEG cap, which consists of multiple electrodes located such that they cover all parts of the head. EEG signals have temporal properties as well as spatial properties. Initially, works were proposed to extract information only through the temporal properties, but slowly, spatial features were also given importance. By extracting the power of the signals, mapping the coordinates of the electrodes on the 2D plane, interpolating the values, we get a sequence of frames which, when analyzed, will help in predictions or extracting valuable insights. The drawback of these methods was that electrodes which were able to communicate in the 3D space were misrepresented when projecting them on the 2D plane. Looking at figure A, when the electrodes are in the 3D space, you will notice that the two electrodes pointed at have a common plane to communicate. However, when these electrodes are projected in the 2D plane, the electrodes are now placed poles apart. This is misinterpreted by the algorithms as entire information is not translated. To combat the problem of loss of information when electrodes are projected in the 2D plane, we map the coordinates of the electrodes onto a 3D grid. The power spectral density values, known as PSD, are assigned to the corresponding electrodes, and these values are interpolated to generate a sequence of 3D frames. The architecture used was a hybrid recurrent 3D convolutional neural network, similar to a video classification task, where 3D convolutional layers were used to extract spatial properties and LSTM layer was used to extract temporal properties. The dataset is prepared by calculating the PSD values for each electrode for the given time, slicing the readings into frames, assigning the PSD values to their corresponding 3D coordinates for each frame, interpolating using Gaussian interpolation. We used a shared 3D convolutional network for input of each frame. The convolutional network was inspired by the VGG architecture. The output of the convolutional network was passed to the LSTM, which passed its output to a fully connected layer. The output was given by applying the softmax activation to predict the most likely output. The optimizer used was Adam, and a dropout of 0.5 was used to combat overfitting. We trained 3 models to test our hypothesis. Model 1 uses our proposed 3D convolutional network for a grid of size 16 by 16 by 16. Model 2 uses 2D convolutional network for grid of size 32 by 32. Model 3 uses 2D convolutional network for grid of size 16 by 16. Observing results, we see that Model 1 outperforms Model 2 as well as Model 3 by 4% in the test set and 6% in the validation set. We also observed that changing the size of the grid does not have a very large difference as long as the coordinates are not overlapping. Through this work, we were able to show that the electrode values, when placed in the 3D space, are better interpreted as no loss of spatial information occurs. Our work aimed at showing the benefit of using 3-dimensional representation of EEG signals. This approach can be further integrated into any state-of-the-art methods like EEG-NET and CRAM to further increase the accuracy of the model.